Hello? Okay. Ooh. So I'm Monica, and today I'll be presenting the first part of my PhD project titled looking at the development of speech-auditory processing during the first year of life. And so for the project, we're generally interested in how infants process acoustic information to process speech sounds. And so we know that infants become attuned to their native language during their first year of life, and a classical study from the 80s shows this, where they presented 6-month-olds who were learning English, 10-month English learning infants, and English-speaking adults with an English native contrast by Da, and a Hindi non-native contrast that isn't native to English. And what they find is that 6-month-olds are able to discriminate both the native and the non-native contrast, whereas 10-month-olds and adults are only able to discriminate the native contrast, but not the non-native contrast. And so given this, we want to know the interplay between this type of native language attunement and any type of attunement to acoustic cues in their native language. And so when we think of the link between the auditory and speech mechanisms, we wonder whether infants process the complex acoustic information of speech signals in the same way that adults do, given their exposure to the language and also their developmental maturity. And we know that the auditory cortex continues to develop throughout adolescence, as you can see in the figure, from the axons in the auditory cortex. And we want to know how infants and adults differently or similarly process information in the speech signal. And so in order to study this, we use what's called a psychoacoustic approach. And so with this approach, we use models that decompose the speech signal in a way that's biologically plausible. And so our cochlea has around 3,500 cilia in the basilar membrane, each of which corresponds to a specific frequency range. And so here you have an image of an unrolled cochlea, and we can see that from the base to the apex, cells respond from frequencies as high as 20 kilohertz, all the way down to as low as 20 hertz. And so with our model, we can conceptualize these inner hair cells in terms of a series of narrowband filters. And in our specific model, we use a model that has 32 filters, which decomposes a sound in a way that mimics cochlear processing. And so this part is what we refer to as spectral decomposition. And so once we've decomposed the spectral information of our sound into our 32 narrow bands, we can take each of these 32 bands and decompose it even further into its temporal components. And so for our temporal components, we can extract the FM, so in blue, which correspond to the fastest frequency modulations in the signal, and the AM, so the amplitude modulations, which are in red, and this corresponds to the overall gradual envelope fluctuations of the signal. And the AM can be even further broken down into its faster and to its slower components. And so the fast AM, and this corresponds to the full envelope that you see in red, and the slow AM would be a more filtered, smoothened version of this envelope. And so we want to know, given what we know about native language attunement between 6 months and 10 months of age, we want to know whether it's possible that listeners learn to rely more on certain acoustic cues that are relevant to the native phonology of their native language. And so just to give you an idea of the information that different cues contain, here's a sentence with just the envelope present. And it might be kind of loud.
A boy fell from the window.
Okay. And then this is the same sentence, but with only the FM present. This is the fastest frequency modulation. Okay. And so, as you might have noticed, and as studies have shown, FM corresponds more to the pitch and the melody, whereas AM corresponds more to the syllabic rate and is sufficient for adults to identify speech in quiet environments. And so our study, we're interested in the role of the FM, the fast AM, and the slow AM. And so as mentioned before, we'll be taking speech sounds, decomposing them spectrally, and then temporally, and then summing up the 32 bands to get what we call our vocoded speech signals. And so in our first condition, we call this the intact condition, we break our speech signal into the 32 bands spectrally, and then temporally decompose them into the AM and the FM, and then sum them back up to create the vocoded signal. Our second condition is the fast condition. So here we want to understand the relative importance of the FM. And so we'll be presenting stimuli with the FM replaced with a pure tone in each band to determine whether or not we elicit a similar or different response to the intact signal. And so our third condition is our slow condition. And so we're, in addition to degrading the FM, we also degrade the amplitude modulations such that only the slowest modulations below 8 hertz are present. And so by doing this, we can see whether the fast amplitude modulations play a role in the processing of our stimuli. And so previous behavioral studies have shown that adults rely on slow AM cues below 8 hertz for consonant discrimination tasks in quiet environments. And whereas 6-month-olds rely more on the fast AM modulations for consonant discrimination. We also know that 10-month-olds, as it pertains to their native language, are attuned to the FM, the frequency modulations, in their native language. And while our study is an EEG study, and we expect that based on what we know about language attunement, we would expect that 10-month-olds would trend toward adult-like patterns. And so we want to know how infants' auditory system neurally processes the acoustic modulations of speech signal to detect consonants. And so specifically, we're using EEG to test the effect of age in 20 6-month-olds, 20 10-month-olds, and 20 adults. And we're looking at the effect of temporal degradation, as I mentioned, with the 3 vocoder conditions, and the effect of French native and non-native consonant. And how we're measuring these are using cortical auditory evoked potentials, CIEPs, and these represent automatic cortical processing of the auditory signals, and they're characterized by a positive peak at about 100 milliseconds post-onset, followed by a negative peak, and followed by another positive peak at around 200 milliseconds post-onset, also known as the P1 and 1P2 component. And some benefits of using this method are that you can use it without presenting any pauses between sounds, and this has been studied in children and adult populations with hearing implants, and also children with cochlear implants. So this gave us a good idea that change detection would be able to be measured in response to our vocoded speech signals. And very concretely, these are our stimuli. So we have 2 consonants, 2 VCVs, vowel consonant vowels, that occur natively in French, aba, apa, and then apa. Pa is the non-native English aspirated consonant, and this is what they sound like in our intact condition with the AM, the FM, and the AM preserved.
Aba, apa, apa.
Okay. And then we present these 3 consonants in our fast condition, where we have the full envelope, but the FM is degraded.
Aba, apa, apa.
And finally, in the slow condition, so where we have only the modulations below 8 hertz preserved. And so we present each of our conditions in 3 blocks. So one block corresponds to one condition. And in each block, we have 200 presentations of each consonant. And each block lasts 7 minutes, and they're presented continuously as VC, VC, VCs, etc. So it's something like a, ba, pa, pa, etc. And so in total, our experiment lasts 21 minutes, and we measured CAEPs after the onset of each consonant, and we compared these responses between vocoder condition for each age group. Okay, and these are our results. So what you're looking at are results comparing our 3 conditions for 6-month-olds, 10-month-olds, and for adults. And so here what we see are the comparisons of our intact versus fast condition and our fast versus slow condition. And so we use cluster-based permutations analyses, and what you see in green are the significant time windows where we found significant clusters, significant differences between our 2 conditions. And what's interesting here is that in all 3 age groups, we see a sensitivity of FM degradation. So right here, when you compare intact versus fast, we see a significant sensitivity of FM degradation. But when you look more closely in terms of the consonant types, what we see are 2 distinct patterns between 6-month-olds and 10-month-olds. So we see that 6-month-olds show a sensitivity to FM degradation for aba and apa, but not for apa. And there's no further sensitivities to fast AM degradation, whereas 10-month-olds only show sensitivities for the non-native aspirated consonant, apa, for both FM degradation and additional fast AM degradation. And adults, so while we do see a similarity between adults and 6-month-olds for aba, overall, adults and 10-month-olds are more similar than 10-month-olds and 6-month-olds in the responses. And so overall, when comparing 6-month-olds and 10-month-olds CAEP patterns, we see similar overall effects of FM degradation, but when dividing consonant by consonants, the 2 age groups, we find that they show distinct patterns according to our results. And when comparing 10-month-olds and adults responses, what we see is contrary to behavioral results. So we would have expected that 10-month-olds and adults show sensitivity to fast AM degradation in addition to FM degradation, but this is perhaps due to a different level of processing. But 10-month-olds and adults, we see that they both show sensitivities to degradation for the non-native apa. And one thing to consider is whether this is an effect of language or simply the acoustics of aspiration. And we're currently in the process of running acoustic analyses to get more information on this. Okay, thank you.